I'm Colin Eadington. I'm a postdoc in Linda Griffith's lab here at MIT.
And my name is Iris Lee. I'm a technical assistant in Linda Griffith's lab at MIT.
And we're working on the Human on a Chip project.
So, Human on a Chip project is a project that's funded by DARPA, and which is aimed at incorporating 10 different organ on a chip models in an interacting circuit to mimic the human physiology. We're basically taking the essential parts of each organ and interconnecting them together.
Any disease that you have is not a disease of just the brain or just the liver or just the gut. It's interacting with all of your organ systems, even if it's primarily affecting one of them. And so you really can't truly understand and treat a disease properly until you understand its effects on the whole system. Especially in the case of the brain, research has really lagged behind because we lack a lot of the in vitro models to generate systems that accurately represent like Alzheimer's and Parkinson's. A lot of these things are very chronic diseases, but typically when you study the brain in vitro studies, you do short experiments, a week or two maybe at the most. But being able to take experiments out for much longer and allowing this interaction with the other organ systems of the body, which are becoming more and more evident as an important player in some of these diseases, allows us to get a much better picture of the disease itself and more accurately develop treatments for it for humans. What you see in the image behind us are neural progenitor cells, which are the precursors to neurons and astrocytes that make up the brain. They've been seated on a gel matrix that allows them to attach and spread and reorganize. What they've done here is organize themselves into a organoid, or a mini organ that has a lot of the same structure and function as parts of the human brain.
So in the middle, the magenta color is a big massive cell body. We see neuronal projections in green that stem from the cell body, and they form intricate networks and connections throughout the whole image. Okay, what I find the most interesting and exciting about this image and this project is that depending on what cellular cues and mechanical cues that we give to the cells, they behave and organize themselves in a very different manner.
So I think the most exciting part of this project is the potential to understand diseases that have been really difficult for us to study in the past. Personally, I have a lot of family members that have Alzheimer's, so I'm very excited that we might have a new, more complex brain model, in addition to things like gut and liver that have also shown to be impactful in Alzheimer's disease. And so I think we could really understand a lot more and move the field forward towards a treatment that has been so elusive in the past. We need to get more of you.
No!
It's going to look like I'm just doing all the talking
good.
while you're sitting here. No, no.
Okay. Oh, this is so awkward! I can't do it!
You'll get better.